Hello everyone, my name is Claudia Bland and I am currently a master's student at Purdue University, and I will be discussing an experiment we conducted to assess the tolerance of selected populations of Palmer amaranth from Indiana to differing rates of dicamba. Palmer amaranth has been identified as one of the most problematic weeds in many cropping systems across the country. Uncontrolled populations can lead to devastating yield losses of 78 and 91% in soybean and corn cropping systems, respectively. Populations have currently been identified in 28 states, and environments are suitable in many additional states for invasion. Throughout the country, different biotypes have been identified exhibiting resistance to 8 different sites of action. Many of these biotypes exhibit multiple resistance mechanisms, making them even more difficult to manage. Specifically here in the Midwest, Palmer has been identified in nearly all crop-producing states. The presence of Palmer in the Midwest is concerning to many growers due to its wide germination window and its ability to reproduce well in non-native environments. In the Midwest in 2013, the germination window of Palmer amaranth was observed to be from May until October. This overlaps with about 75% of the growing season, so there can be continuous competition between Palmer amaranth and the cash crop, which can cause decreased crop quality and yield losses. Fecundity of Palmer, even in non-native environments, is also a concern. Research conducted in Indiana and Arkansas in 2013 and 2014 looked at the inflorescence and growth of Palmer amaranth populations from Arkansas, Indiana, Mississippi, Missouri, and Nebraska, and showed that all non-native populations not only survived, but accumulated significant biomass and produced seed in both Arkansas and Indiana. This shows that even when removed from their favored environment, Palmer amaranth populations can pose a significant amount of competition on the cash crop. Looking more specifically at Palmer amaranth in Indiana, it was first identified in 1955, but did not pose a serious threat to cropping systems until 2011. These more problematic populations were observed in 2 northwestern Indiana counties, Posey and Vanderburgh, and already exhibited resistance to glyphosate. The introduction of Palmer amaranth to the state of Indiana was through contaminated cotton hulls from the southern states used as a fiber source in cattle feed. After digestion, manure from these cattle was spread onto the fields for fertilizer, which introduced the Palmer amaranth seeds to optimal growing conditions. Now it has spread to 24 counties throughout the state by human interaction, such as contaminated farm equipment, or natural causes, such as wildlife. Biotypes that are from Indiana, according to that same study from Arkansas and Indiana, that are established early have higher seed production and biomass accumulation than those established later. Indiana populations also require fewer growing degree days to reach 95% inflorescence and 20 centimeters in height. These populations have adapted to establish and reproduce quickly, which can be problematic when coupled with their enhanced ability to develop mechanisms of resistance to herbicides. In Indiana, Palmer amaranth has developed resistance to 5 sites of action. The newest addition to the list of resistance mechanisms of Palmer amaranth nationally is the Group 4 synthetic auxin herbicide class. Research at the University of Arkansas suggested some populations showed reduced sensitivity to dicamba. Additionally, in 2019, a population in Kansas was confirmed resistant to 2,4-D. This new addition poses a threat to the use of synthetic auxin-tolerant crop varieties, just as many growers are beginning to adopt these varieties into their programs. In the research done by the University of Arkansas, populations tolerant to a field application of dicamba were selected for subsequent screening in the greenhouse. Survivors were then propagated and screened again for 3 continuous generations. In this table are the GR50 and GR90 values, followed by their 95% confidence intervals. It was concluded that as plants were continually propagated after survival, they increased in tolerance to dicamba, and the P3 population had the highest tolerance. In Indiana, there has been no confirmed resistance to the synthetic auxin class. However, low levels of tolerance were observed in a whole plant dose-response assay conducted at Purdue University in 2013 and 2014 by Doug Sponhorst. The goal of his study was to characterize 41 different populations from Indiana for sensitivity to 9 post-herbicides. With the known resistance to ALS herbicides in Indiana, it was no surprise that 100% of populations were controlled. There was also no surprise that over 80% of populations were resistant to glyphosate, as even the first populations identified in Indiana were resistant. With the observed resistance to mesotrione and HPPD herbicide, At over 90%, we can conclude that since HPPDs and synthetic auxins are both metabolism-based herbicide classes, that sensitivity to synthetic auxins is plausible in these populations. 5 populations showed very low levels of tolerance to a field use rate of dicamba. Further investigation was needed to determine if these populations were showing reduced sensitivity to dicamba. 4 of these populations plus a sensitive check were selected for subsequent screening. Based on the conclusions made by Sponhorst, we hypothesized that the 5 selected Palmer amaranth populations from Indiana would exhibit differential responses to foliar applications of dicamba. Our objective was to evaluate these 5 selected populations of Palmer amaranth to differing rates of foliar applications of dicamba and compare them. To evaluate the response of these populations to dicamba, a whole plant dose-response assay was conducted in the greenhouse at Purdue University in West Lafayette, Indiana. This assay was conducted as a 2-factor factorial, the first factor being population and the other being dose of dicamba, in a randomized complete block design with 8 replications. Data were analyzed in R using the nonlinear regression via the DRC package, and the GR50 and GR90 values were calculated for each population using the dried biomass data. Seeds were gathered from storage and scarified with a 10% bleach solution to increase germination rates and then sown into flats with moistened potting mix. Undocumented research from Purdue University showed better herbicide efficacy with the Amaranthus species when using a 1-to-1 ratio of field soil and sand compared to a 2-to-1 ratio of potting mix and sand. So when seedlings reached the first true leaf stage around 72 hours after planting, they were transplanted into 10 centimeter square pots filled with the 1-to-1 field soil sand mixture. Once plants reached the 6 to 8 true leaf size, differing doses of dicamba were applied at 140 liters per hectare, and transplants were fertilized weekly. Tolerance was assessed in 2 ways, including visual estimates of control and counts of live and dead plants to assess survival rate. Visual estimates of control were taken at 14 and 21 days after treatment, and live plants were counted at 21 days. Estimates were made using a percent scale where 0 represented no control and 100 represented total plant death. Above ground biomass was collected following the 21-day rating, and following biomass collection, samples were dried for 72 hours and dry weights were measured. Now let's dive into discussing the results. For a more objective measurement, we compared biomass reduction of each plant within a population to the mean of the population control, and here is what we saw. In this graph, rate is represented on the x-axis and biomass reduction on the y. No differences were observed between the populations, including the sensitive check, which is displayed here as population 1. Since the sensitive check produced similar results to the other populations, we conclude that we did not select a sensitive enough population for comparison, and in future studies, a more sensitive population will be needed in order to compare. Looking only at biomass data can be misleading with auxin herbicides due to the increased production of callus tissue caused by the application. So in addition to biomass, visual control estimates were analyzed. Looking first at the dose-response curve for the visual estimates at 14 days after treatment, we have rate on the x-axis and percent control on the y. This graph shows us the same thing that we saw in our biomass curve. None of these populations showed differential responses. At the lower rates, there is a larger spread in the data, which is making it difficult for the dose response to sort out differences. Moving into the percent control at 21 days after treatment, we again have rate on the x-axis and percent control on the y. We did observe increased injury at all rates, but again we saw no differences between the populations, including the sensitive check. The GR50 and GR90 values were calculated with the dry above ground biomass data, and a t-test was conducted in order to compare differences in these values of all the populations. Values are followed by their standard error. Although no differences were observed, the GR90 values show that some of these populations may be getting close to surviving an application of the field use rate. Specifically, population 3, although it has the highest standard error, shows it is closest of all the populations to being able to survive the labeled field use rate. Further investigation is needed to confirm this observation. Looking back at values from the study in Arkansas in 2017, we can compare the GR50 and GR90 values to our study. The GR50 values from our study are way lower, but our GR90 values align closely with the P1 and P2 generations. We can conclude from this that as our selected populations reproduce, the GR90 values may increase to a level equivalent to what was deemed to have reduced sensitivity in the Arkansas study. The regression analysis of biomass and visual control estimates show no differences. However, we did observe some trends in the survival rate between populations. In this bar graph, the rate is represented on the x-axis and the number of survivors on the y-axis. In the lower two Dicamba rates, all populations behaved similarly except for population 5, which showed a 75% survival in the lowest rate compared to 100% survival in all other populations. As we get into the next two Dicamba rates, 114 and 227 grams, the populations began to show more variance. Again, population 5 was the most sensitive, having only 3 and 1 surviving plants respectively. The other populations, excluding the Czech, were at least doubled in the number of survivors compared to population 5, and we were seeing survival rates between 25 and 88% across both rates. This shows that some plants are developing tolerance to these lower rates, which could be problematic as they continue to reproduce. Finally, as we look at the highest rate, we see that at least one plant from all populations except the sensitive Czech survived. That means every population has at least a 12.5% survival rate, which would be considered a failed application in the field. From this, we can conclude that all populations have potentially problematic plants surviving application, but will require further investigation to confirm. Based on the dose-response data, there are no differences among populations, so we would reject our null hypothesis. However, with the survival rates we observed, we conclude that these populations are problematic as we are not seeing total plant death at the highest rate. Therefore, further investigation into these selected populations is needed to confirm these survival rates. Based on what we observed in this study, Dicamba is not an effective control tactic for Palmer amaranth when used alone. These populations were only marginally controlled, meaning resprays would be necessary for total control in a field situation. An effective management strategy for Palmer amaranth requires more than just Dicamba. Furthermore, to understand why these plants are showing these low levels of tolerance, investigation of the survivors by propagation and respray is necessary to determine the mechanism of resistance. As mentioned, continual screening of populations of Palmer amaranth for differing levels of tolerance to auxin herbicides is important in order to monitor tolerance levels across the state. Propagation of surviving plants from this screen should also be done to further investigate tolerance levels of these selected populations from Indiana. As these levels of tolerance begin to rise, there is a need to look into how to maximize the efficacy of Dicamba applications on current systems, which I will address throughout my degree by manipulating spray parameters and looking at differing levels of tolerance of Palmer amaranth populations to these changes. With that, I would like to thank the current staff and graduate students in the Purdue Weed Science Lab for their help and support, as well as Doug Sponhorst and past Weed Science graduate students for their contributions to this project. Thank you for your attention, and I would be happy to take any questions at this time.